Hello friends, welcome to some biology class. Friends, today we shall discuss about the lectin pathway of complement activation. Like the classical pathway, the lectin pathway of complement activation proceeds through the activation of C3 convertase, which is composed of C4b and C2a. However, instead of relying on antibodies to recognize the microbial threat and to initiate the activation process, this pathway uses lectins. Lectins are the proteins that recognize specific carbohydrate components that are found primarily on microbial surface as its specific receptor molecules. Mannose binding lectin MBL, the first lectin demonstrated to be capable of initiating the activation process, bind a close-knit array of mannose residues that are present on the surface of many bacteria, fungi, and even few viruses. In addition to mannose binding lectin also recognize other carbohydrate components such as N-acetyl glucosamine, D-glucose, and L-fucose, which are present on microbial surface. All these sugars present their associated hydroxyl group in a three-dimensional array. Mannose binding lectin is constitutively expressed by the liver. It is structurally similar to C1q component of the complement system. It belongs to a subclass of lectin known as collectins. In addition to MBL, another family of proteins known as picoline has also been identified as the additional initiator of the lectin pathway of complement activation. Three different picolines such as L-picoline, H-picoline, and N-picoline recognize and bind with specific carbohydrate components which are present on microbial surface. In serum, mannose binding lectin is associated with MBL associated serum protease known as MASP. Three different mannose binding lectin associated serum proteases such as MASP1, MASP2, and MASP3 have been identified. All the sugars including mannose present their associated hydroxyl groups in a particular three-dimensional array. Mannose binding lectin is constitutively expressed by the liver. It is structurally similar to C1q component of the complement system. It belongs to the subclass of lectin known as collectins. An additional family of proteins known as picolines, which is structurally related to collectins, have been identified as the additional initiator of the lectin pathway of complement activation. Three different picolines have been identified. They are L-picoline, H-picoline, and N-picoline, and each of them bind specific carbohydrate components. In the serum, mannose binding lectin is associated with the MBL associated serine protease known as MASP. Three different MBL associated serine proteases have been identified. They are MASP1, MASP2, and MASP3. This figure shows the initiation of the lectin pathway. This is a microbe, and on its surface there are a number of polysaccharide antigens. The mannose residues that are present in those polysaccharide antigens are recognized by the mannose binding lectins, And these are the molecules of MAsP1 or MAsP2. MAsP2 is structurally related to the serine protease C1s and cleave the complement components C4 and C2, thereby leading to the generation of the C3 convertase, which is composed of C4b and C2a. The lectin pathway use all the same components of the classical pathway with the single exception of C1 complex. In the lectin pathway, soluble lectin receptors replace the antibodies as the antigen recognizing component, and MASP proteins takes the place of C1R and C1S in cleaving and activating C3 convertase. Once the C3 convertase is formed, remaining reactions of the lectin pathway are same as that of the classical pathway. Like the classical pathway, in lectin pathway also, the C5 convertase is C4b2A3b. The lectin pathway is considered as a part of the innate immune system because it do not rely on antibodies to recognize the microbial threat and to initiate the activation process. Instead of it, it uses specific proteins known as lectin. So friends, this is about the lectin pathway of complement activation.